All right, guys, today let's talk about dihybrid crosses. Now, if we look at the word di means two, and then a hybrid is going to mean like different things. Like think of a hybrid car, it's gas and electric. So instead of just looking at one trait, now we're going to look at two. So a dihybrid is a genetic cross that shows two distinct genes. For example, like if we look at height, and we look at eye color. That's two distinct traits. Those are two distinct genes. And sometimes genes tend to get inherited together, especially if they're really close to each other on the chromosome and we do crossing over. Now, if we look at height, specifically, we can still just do dominant recessive. So we'll pretend that tall is dominant and recessive is short. And then, of course, if we look at eye color, we'll pretend that brown eyes are dominant to blue eyes, right? And then from our regular monohybrid crosses, the ones we've been practicing, you still get to do your genotypes. So if you have two big A's, remember you get one from mom, one from dad, that means they're going to show that tall trait because they have at least one dominant allele. And if you get a big allele from one parent and a small allele from the other, you're still going to show that dominant trait because you have at least one big allele. And then if you have two little alleles, that means you're going to show the recessive trait, which will be short for this height one. Now, if I look at the other one, so remember eye color, brown is dominant to blue. If you get two big alleles, one from each parent, they'll show brown eyes. If you get a big allele or a dominant allele and a recessive allele from a parent, that means you're still going to show brown eyes because you have that dominant trait. And then if you get two recessive alleles from each of your parents, then that means you'll show the recessive trait, which is blue eyes. And so we can start having all sorts of combinations since we have two genes, right? Or two traits to look at. So if we look at this one, like you could get two big alleles from height and two big alleles from eye color from your parents. So this person would be tall with brown eyes because they have at least one allele or one dominant allele from each trait. You could have a heterozygous for height and a homozygous dominant for eye color, which would still show tall with brown eyes because you have at least one big letter for each trait. You could be heterozygous for both of these traits, but you'd still be tall with brown eyes because you have at least one big letter. And then you could be homodom for tall, and you could be hetero for eye color, which would still show you tall with brown eyes. Or you could have this combination. You could have two recessive alleles for height and then two dominant alleles for eye color. So that means you'd be short with brown eyes. Or you can get two recessive alleles from your parents for height and a heterozygous from your parents. So it's still short with brown eyes. And then if we move on to this one, you could have a dominant trait for height and recessive for eye color. Because homodom will show dominant, hetero will show dominant, and then two littles will give you recessive, which means you'd be tall with blue eyes. Or you can get recessive for both traits and short with blue eyes. And so now instead of having just two options for phenotypes, you get to have four options because you get to have the different the different combinations, sorry. But that's all well and good, but what we want to do with genetics is we want to use a Punnett square to help predict all of these offspring. So the first thing you need to do is get your two parents. And we're going to say mom is heterozygous for both traits, and we're going to say dad is hetero for height and homorecessive for eye color. And what we have to do once we find out our genotypes for our parents, we have to figure out the gamete possibilities. So we have to FOIL this sucker. And you guys have heard of FOIL before, because that FOIL just means first, outside, inside, last. And so we're going to FOIL each of the genotypes. So if we look at mom, let's do the first of each letter. So that would be a big A and a big B. And so here in one of my gametes, because remember we get to make 4 gametes, 4 egg and 4 sperm. So in one of my gametes, I'd have a big A, big B. Now let's do the next one. Oh, that's outside. So I'll do the outside of each letter. So I'll have big A and little b. So now I have big A and little b in a gamete. Then I do the I, which is the inside of each letter. So little a, big B. And so that would go in a possible gamete, possible egg, little a, big B. And then you have to do L, which is last of each letter, which would be the little a, little b, and that would be in a gamete. So I figured out my gametes for my mom. Now we have to do my dad. So we do first of each letter, which would be A, big A, little b. So that would go in a sperm. I have to do the outside of each letter, which would be big A, little b. That will go in a sperm. I have to do the inside of each letter, which would be little a, little b. That would go in a gamete or a sperm. And then I do the last of each letter, which would be little a and little b. There you go. So now we've foiled out our parents, so we can set up our Punnett square. And so what we do is we just look at our mom gametes, like these ones, and we put one gamete over each box. So now instead of having 4 boxes, I have 16. So I'd put big A, big B, big A, little b, little a, big B, little a, little b. That's where I just got those. So I just took those 4 gametes that I foiled out and put them over the boxes. Now I'll do the same for Dad. Here are his 4 gametes. So I'll put big A, little b right here. And then the next one was big A, little b. And then little a, little b, little a, little b. And now I just set it up like a regular Punnett square. I just put all the letters, but I keep my letters together. So I'll have big A, and then big B, little b. Big A, little b, little b. Big A, big B, little b. Big A, little b, little b. Okay, now I do the next row. So big, big, little, big, little, big, little, big, little, little. Now I do the next row. So big, little, big, little, big, little, 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 big, little, little. And now I do my last row. Big, little, big, little, big, little, little, big, little. All right, so now all of those boxes that have those dihybrids, those are possible offspring combinations. So now we just get to go through and count to see how many are both dominant or will have both of the dominant traits, or how many will have dominant of one trait and recessive another, or recessive of that first trait and dominant of the other, or 2 recessive. So I just count boxes. So to look for dominant of both traits, I have to have at least 1 big letter in each one. So let's go through. All right, this one has a big A and a big B. So there's 1. Not this one, because he has a big A, but 2 little b's. This one, big A, big B, so there's 2. Not that 4th one. This one has a big of each, so there's 3. Not this one. This one does, so that's 4. Not that one. This one does, so that's 5. Not this one, not this one, not this one. This one does, so that's 6. And no. So I have 6 of my 16 offspring will show the dominant trait for both of them. So that means they'd be tall with brown eyes if we were going off of that first example. So now let's look at dominant recessive. So at least 1 big A and 2 little b's. Well, I can skip that first box. All right, so he has big A and 2 little b's. So there's 1. Skip that box. Big A, 2 little b's. There's 2. Skip this one. Big A, 2 little b's. There's 3. Skip that one. Big A, 2 little b's. There's 4. Skip that one. Big A, 2 little b's. There's 5. Nope. Not a big A. So I have 5. Not that one. Big A, 2 little b's. There's 6. And not that one and not that one. So this one is also 6 of my 16 offspring will show the dominant trait of one, but recessive of another. So this would be tall with blue eyes. Now let's look at recessive for the first gene, but dominant for the other. So 2 little a's and at least 1 big B. All right, that first row is done. That second row is done. this one, because I have 2 little a's and 1 big B, so there's 1, not him. Those 2 are done. And this one, 2 little a's and 1 big B, so there's 2. So 2 of my 16 offspring will be short with brown eyes. And now I can do my last one. That first row is done. Whoa, let me do that. That first row is done. That second row is done. This one, 2 little, so there's 1. And then there's another one, 2 little. So that would also be 2 of my 16 offspring will show both recessive traits. All right, so that's how you do dihybrids. Dihybrids mean you have 2 distinct traits. You have a bunch of different gamete possibilities. Still follows the regular rules. If you have at least 1 big of each letter, they'll each show the dominant trait. If you have 2 little letters of 1 trait, then that means they'll show the recessive for that one. To set up a Punnett square, you have to take your parent and you have to foil their genotype with the first outside, inside, last. And then once you foil it, you can set up your Punnett square and find out your gamete combinations. All right, hope that helps.